Assalamu alaikum. Welcome to my new video. In this video, we can discuss about gene and its types. So, first of all, we will discuss about genes and then we can discuss its types. So, the history of gene is the heredity unit, which we call gene, now was proposed by Mendel, and they give the name elementin. Mendel gave the name elemental, and then later on, William Johnson introduced the term gene for the basic unit of heredity in 1909. In 1909, William Johnson gave the name it gene. So, what the gene word means? The gene word is derived from the Greek word genos, which means generation or birth. What is the definition of gene? So the definition of we can define gene like that the gene is a specific segment of DNA that give instruction for polypeptide synthesis, or DNA or gene is a segment of DNA that responsible for inheritance and expression of particular character. We can define gene like that gene is a specific segment of DNA that give instruction for polypeptide synthesis. Or we can also define gene like that gene is a specific segment of DNA that are responsible for inheritance and expression of particular character. So it has a lot of definition according to their function and the cell. And the first most important definition or most important function of gene is that the gene is a specific segment of DNA that give instruction or information about protein synthesis or polypeptide synthesis. This is the most important function of gene. And the next important function of gene and definition is that the gene is a specific segment of DNA that are responsible for inheritance and for expression of particular character. We can also define gene like that gene are basic and functional unit of hereditary information that occupies a fixed position on a chromosome. And we can say that gene is a specific segment of DNA, but in viral genome, we can also say because in virus that only DNA is present or RNA is present. Both DNA or RNA are not present simultaneously. So we can define gene at that time. That we can also define a gene in viral genome when DNA is present. Then we can say that gene is a specific segment of DNA. But in those viruses whose genome consists of RNA, then we can say that gene is a specific segment of RNA that has a function which they perform in viruses. So this is not necessary that we all say that gene is a specific segment of DNA in virus. In viral genome where RNA is present, then we can say that gene is a specific segment of RNA. So this is the history, meaning and definition of gene. Now we can move towards its types. There are a lot of types. There are many types of gene. So we can discuss only 5 types of gene. The first one is housekeeping gene or constitutive gene. So the first step of gene is a housekeeping gene and constitutive gene. What is housekeeping gene and what are constitutive gene? So housekeeping gene are those genes in genome that tend to be highly conserved and evolved slower than other genes, and they are tissue specific genes because mainly their role is in maintenance of basic cellular function, and they are also essential for existence of cells. Why we called it housekeeping gene? Because they code for protein that are constantly required by the cell, and they are essential to cell and always present under any condition. It is assumed that their expression is unaffected by environmental or any experimental condition. They are called housekeeping gene because they are constantly present when they are required or not required, but their presence is constant. They are constantly required by the cell and they are constantly present in the cell. Why they are called constitutive gene? So they are always active, and that's why they are called constitutive gene. And the example of housekeeping gene or constitutive genes are genes for ribosomes. Genes for ribosomes are an example of housekeeping gene because the ribosome genes are constantly being transcribed because ribosomes are constantly needed for protein synthesis. So they are the example of housekeeping gene or constitutive gene. The second type of gene is non-constitutive gene. So what is non-constitutive gene? They are opposite of constitutive gene, as constitutive genes are constantly required by the cell and they are present at any time in the cell. So non-constitutive genes are not always required by the cell and they are not expressed in all cells. They are tissue specific or organ specific and they are not constantly expressed, or they are expressed only when their function is needed. Example of non-constitutive gene is plasmid of bacteria and gene for heat shock protein. Heat shock protein and plasmid of bacteria are the example of non-constitutive gene. Non-constitutive gene are also called luxury genes. So this is the example of non-constitutive gene. The third type of gene is structural gene. So what are structural genes? As the name indicates that they are responsible for the structure of genes. So for structuring the gene. So they are that type of gene, those genes which are required for, which can code for synthesis of specific polypeptide. For synthesis of specific polypeptide, they are responsible for their coding. Example are operator genes because operator genes contain code necessary to begin the process of transcribing the DNA message of one or more structure gene into mRNA. So, in order to transcribe the DNA message of one or more structure gene into mRNA, operator gene contain code and which are necessary for transcribing the DNA. So, this is the example of structure gene. Okay, the fourth type of gene is pseudogenes. So what are pseudogenes? Pseudogenes is a DNA sequence that resemble a gene but has been mutated into an inactive form over the course of evolution. Pseudogenes are former genes that have accumulated mutation and they make them non-functional, but sometimes we called it vestigial genes. And we know that what is vestigial, what the term vestigial means. The term vestigial is used for that thing which is non-functional. So we called pseudogenes the vestigial genes because they lost its function in the course of evolution. In the course of evolution, they lost its function and become non-functional due to accumulation of mutation. So that's why we called it vestigial genes. The fifth type of gene is transposons genes or jumping genes. So as the name indicate, they are jumping genes. So we define transposon genes like that those genes which change its position within the genome, and they are repetitive DNA sequence that have capability to move from one location to another location in genome, and they are also called mobile elements. Transposal genes or jumping genes are important to evolutions because jumping genes can jump around the genome in developing sperm and egg cells. And they can result in mutation and they can also alter the gene expression, induce chromosome rearrangement due to increase in copy number, and they can enlarge genome size. And they can often result from duplication of same genetic material from due to same genetic material. They can jump from one location to another location. So this is known as transposon genes or jumping genes. Okay, in this video we can discuss about gene and its type. We can also discuss about the history of genes, about their meaning, about their definitions, and we can also define gene in a virus. And we can also discuss in this video about its types. So there are many types of genes, I think about 17 or 18 types, but we can discuss only 5 types in this video. We can also discuss the another types of genes in the next video. So see you in the next video. Allah Hafiz.